this is the first time we will revise what we have done in the previous lectures. We have studied about the host cells and the factors of the cloning, and we have explained what was the role of the prokaryotic host cell, what is the role of the E. coli and Bacillus stabilis, and if what are eukaryotic host cells, why are they preferred, then the prokaryotic is host cells, and then we have also explained some host cells and what is the uh, uh, what are who has the strong promoters and post translational modification. Then we have also explained you the Chinese hemisphere ovary cells. So check the previous videos and see what I have explained in all those videos. Now this is the new topic which is called as the vectors, and this is very important topic and you have to understand this. So first of all, we try to understand that what vectors are. So vectors are the DNA molecules which carry the foreign DNA fragment. Yeah, that means that vectors carry the foreign DNA fragment to be cloned. Yeah. For example, if we want to clone this portion of the gene, and we have cut this gene from the DNA, and now this gene is introduced into the vector, and when it is introduced into the vector, this foreign portion of this vector will go inside the host cell, and after going inside the host cell, it will be cloned. It will be cloned. So the vectors are the vehicles which transport this foreign gene into the host cell, and that's why they are called as the vectors. That means they are called as the vehicles. So what are the properties of the vectors? The vectors are self-replicating in an appropriate host cell. That means the host cell need to be appropriate. They will not self-replicate in any of the cell. That means if you will choose any of the cell and you don't know whether this vector will replicate inside this cell, so this cannot be a appropriate host cell. So appropriate host cell is important for the vector to self-replicate. This need to be understood. So there are different types of vectors. We have plasmid vectors, we have bacteriophage vectors, cosmid vectors, and artificial chromosome vectors. We will try to understand all of these in the coming time. So first of all, we will try to see what is the plasmid vector, and how many types of the plasmid vectors are. So plasmid, what are plasmids? So plasmids are extra chromosomal DNA. That means if this is the diagram of the bacteria, and this bacteria is having the circular DNA, and this is the genomic DNA. That is the genomic DNA, that is chromosome genomic DNA of the bacteria, that is also called as a chromosomal DNA. Chromosomal DNA. So along with this chromosomal DNA, there is a circular DNA, that is apart from this chromosomal DNA, there is another type of DNA that is called as the circular DNA, and that circular DNA is extra chromosomal DNA, that is extra chromosomal DNA. So we have in the bacteria chromosomal DNA, and extra to this DNA is a circular DNA, that is called as a plasmid. So you can see in this diagram that this is the chromosomal DNA, that is the nucleolus, where the nucleolus is present in the bacteria, and this is the circular DNA, that is called as the plasmid DNA, and this is the structure, that is this bacteria is having the flagellum, capsule, cell wall, plasma membrane, pili, and cytoplasm, and 70s ribosomes. Now, uh, some of the plasmids are having different shapes, like this is the loop shaped double stranded DNA plasmid vector, and this is the streptomyces tenace circuitous racket end shaped plasmid vector. So it's not necessary that this plasmid vector will be always having the circular shape. It can be in the of other shape. So this is a double stranded circular DNA. Now, is this plasmid always circular in shape? Now we have some exceptions. So exceptions are here, like uh, we have like we have borilla borilla, and we have streptomyces. They are having the linear shaped vectors. They are having also the double stranded DNA, but they are not having the circular but linear DNA. And one is the racket end type, tennis racket end type DNA is plasmid, and another is having the hairpin, loop, and plasmid. So this need to be understood and this need to be remembered. You will not find this information any other video, or nobody has discussed this. As I know, I have checked most of the videos, but to the best of my knowledge, some might have helped you to understand this concept that there are other types of the plasmid DNAs as well, and one the linear exception is the linear type of DNA, that is hairpin or loop shaped DNA, is in Streptomyces borrella and Streptomyces. So this need to be understood. And this plasmid is also self-replicating DNA molecule. Now, almost all the bacteria have the plasmid containing at low copy number. That is, what is the copy number? Copy number is how many plasmids can be. Is this a genomic DNA, yeah, or chromosomal DNA, and how many plasmids can be inside the bacteria cell? It can be one, or it can be four. So it depends upon. Or it to the high copy number is having 10 or 100 per cell. That means in a per cell there can be 10 or 100, it depends. So the size of the plasmid is 1 to 500 kb, and usually the plasmid contributes to the 0 to 5% of the DNA of the bacteria. If we will see that the 100% is the percentage is the DNA of the bacteria from the chromosomal DNA, now this 5% will be of that DNA plasmids. So we have some plasmids that is called the PET28A plasmids. They are the expression vectors, and P means plasmid and ET means expression vector, and specific name 28A is specific and identified in this series. And we have the plasmid GEX41, and this is the glutathione S transferase expression vector, and 40 is a specific identifier in this series. I will explain these two of the vectors, about these two vectors in the coming videos, and I will give you a separate lecture on them. What is this PET? They are the expression vectors. If you want to get the purified DNA proteins, you want to get the proteins in a purified form, then we use the PET28A. And why? And they have the different type of host cell. That's why you will not use these vectors in case of E. coli as a host cell. So we have the different host cell, that is BCL1 host cell there. Now, we have the PUC19, that is plasmid, and University of California, where this vector was designed and identified as 19. And we have PBR. And most of this have taught you that there are two types of the plasmid vectors. One is PBR 322, it is a synthetic plasmid, and PUC19, it is also a synthetic plasmid, but you have to remember PET28A and PGX, that is glutathione S transferase expression vector 41. So this is the name by who has designed this vector, that is BR Bolivar and Rodriguez. These are the scientists' names, and identifying number is 322. So there are 322. Then after this, they have designed another vector that is having the 323 and the 324, and this in the same way PUC 19, 20, 21. And we will first start with the PBR 322, the most common plasmid vector. So this is the most common plasmid vector of E. coli and most popular, widely used as the plasmid vector. So PBR has the DNA sequence of 431 base pairs, and it carries the resistance gene for ampicillin resistance gene, and I will explain you, and tetracycline resistance gene. So what is this ampicillin resistance gene? Ampicillin resistance gene means that if we will clone these vectors inside the host cell, now we can easily give the ampicillin drug, because antibiotic in the media media, and this resistance gene will allow it to grow, because they will show the resistance to the ampicillin. So in the same way tetracycline though, so they will show the resistance to the tetracycline as well as ampicillin. So the reason is why we have kept the ampicillin and tetracycline, and they serve as the markers for identification of clones carrying the vector. For example, what does this mean? One second. These are this is some the trouble happened here, and we will reach directly to this topic. Now, what does this mean? They act as a serve as the markers for identification of the clone. For example, these are the bacterial cells. These are the host bacteria cells. These three and four. In this, we have this plasmid PBR 322. In this, we have this PBR 322. Now, the plasmid has not entered into this cell and they lack these vectors. So, if we will grow the host cell, these media into the ampicillin antibiotic. The medium contains ampicillin antibiotic. These cells who are not having the PBR with ampicillin resistance gene will die because this will be sensitive to them, and those cells which were having the vector inside them will live because this ampicillin will show resistance to the ampicillin resistant gene will show resistance to the, and it will not be sensitive to these cells, and we will easily understand as only those cells who live there are having the vector inside them. So this is very easy to understand that they are serve as the identification markers or clones. Now, the plasmid's unique recognition site this plasmid is also having the unique recognition site for the action of restriction endonucleases. So, what are those sites? They are, you can see, they are ECO-R1 site where the ECO-R1 resistance the ECO-R1 restriction endonuclease is cut. Then HIN3, BAM-H1, SAL1, and PST1. So they will cut at these sites, and we can use all these restriction endonucleases so that we can cut this vector and insert the gene of interest into it. Now, the plasmid was named is because after the Bolivar and Rodriguez started to form this, and this you can say that the plasmid is having the other things as well as with this ampicillin tetracycline and other restriction endonucleases with them, and we are mostly learning about four of these types of restriction endonuclease in order to remember and make it easy for you. Now we have the PUC. PUC is, you know, that it's the Plasmid University of California. It is having 268 base pairs. You have to remember that this PBR is having the 4 4361 base pairs, and it is having the 268 base pairs along and contains ampicillin resistance gene. So ampicillin resistance gene is, and a segment of beta galactosidase gene, LAC-Z gene of lactose operon of E. coli under the control of regulatable LAC promoter. So LAC1 repressor, LAC1 gene that produces the repressor protein that regulates the expression of LAC genes from the for the LAC promoter. So what is this LAC1? That LAC1 is a gene that produces a repressor protein. Repressor proteins repress the protein synthesis and regulate the expression of LAC-Z gene from the LAC promoter. So this is very important. It is having the LAC-Z gene. It is having the LAC1 gene repressor, mostly asked in the questions, and multiple cloning sequences, ampicillin resistance, and origin of replication. A short DNA sequence with many unique cloning sites like ECOR1, SAC1, KPN1, ZAMA1, SAMA1, and BAMH1, ZABA1, SAL1, and HINC second, ACA second, BSP second, PST second, P. These are the restriction endonucleosides. That is why we are saying that it has multiple cloning sequences and having the multiple restriction endonucleases, sites for endonucleases, which can multiply the cloning sites, and that's why multiple cloning sequences or polylinkers are there, and the origin of replication for PBR3 from PBR 322. So most of many sometimes you may be asked why from this origin of replication has been introduced in PUC 19. It is from the PBR 322. And what is this multiple cloning sequences? It is having these number of the restriction endonuclease sites. That means that these we can use these number of the restriction endonuclease to cut this. So you can see this polylinker cloning site having all these restriction endonucleases, and ampicillin resistant gene is also having some of the restriction endonucleases. Now, what is this ampicillin? Ampicillin, what is the role of this ampicillin antibiotic? It inhibits the cell wall formation, inactivates the beta lactamase. What is the role of the hygromycin? Hygromycin blocks translocation from amino acyl site to the peptidyl site and inactivates by the phosphotransferase. What is the role of the kanamycin? Kanamycin binds to 70S subunit of ribosome. Ribosome is having the two subunits, larger and smaller subunits. In that, there is 30S subunit and prevents the translocation from amino acid tRNA site to peptidyl site and inactivated by the phosphotransferases. What is the role of neomycin? Neomycin also binds to the 30th subunit and inhibits the protein synthesis inactivated by phosphotransferases. What is the role of streptomycin? Streptomycin blocks the protein insertion complex formation and causes misreading during the translation and inactivated by their phosphotransferase. So they can be inactivated by phosphotransferases. Tetracycline prevents binding of amino acid tRNA 30th ribosomal subunit and restriction gene encoded inner membrane protein that passes the antibiotic out of the cell. That means that it causes the efflux of the antibiotic and blocks the passage of antibiotics through the cell. So this all for